There are moments in paleoanthropology when a fossil does not simply sit within a timeline, but begins to bend that timeline around itself, forcing researchers to make decisions that are not entirely dictated by the bone, but by expectation, by prior models, and by the subtle pressure to make disparate pieces of evidence align into something coherent, something recognizable, something that looks, at least in outline, like a plausible ancestor. One of those moments began in the early 1970s, with the discovery of a skull along the eastern shores of Lake Turkana, a specimen that would later be known as KNM-ER 1470, and which would become the centerpiece of what is now called the Homo rudolfensis controversy, a debate that does not merely concern the classification of a single fossil, but the very process by which fossils are reconstructed, interpreted, and ultimately transformed into evolutionary narratives. At first glance, the skull seemed to offer something extraordinary, a combination of features that appeared to bridge a gap, with a cranial capacity often cited around 750 cubic centimeters, significantly larger than typical Australopithecines, yet paired with a face that, in its initial reconstruction, appeared relatively flat, almost modern in its verticality, creating an image that was both familiar and unexpected. It was this combination, a larger brain case and a less projecting face, that gave the fossil its initial appeal as a potential early member of the Homo genus. And yet from the very beginning, there were disagreements about how the skull should be assembled, disagreements that would prove to be far more consequential than they first appeared. The fragments of KNM-ER 1470 did not arrive as a complete intact skull, but as a collection of pieces that required careful alignment, interpretation, and inevitably a degree of judgment, particularly in determining the angle at which the face met the cranial base. This angle, seemingly a small technical detail, would become the fulcrum upon which the entire classification rested, because a slight rotation forward or backward could dramatically alter the perceived flatness of the face, the height of the mid-face, and even the apparent size of the brain case. It is here that the role of expectation begins to emerge, because different reconstructions produce different organisms, not in a trivial sense, but in a way that could shift the fossil from one species category to another. According to accounts from those involved in the original work, including Alan Walker, the initial reconstruction presented a more projecting face, one that aligned more closely with Australopithecine morphology, a result that did not sit comfortably with the emerging idea of a larger-brained, more human-like ancestor at that time. The skull was subsequently reassembled with the face positioned more vertically beneath the brain case, producing a markedly different appearance, one that seemed to support the classification of Homo rudolfensis. This was not merely a cosmetic change, but a structural one, altering the spatial relationships between key anatomical landmarks and crucially affecting estimates of cranial capacity. As later critiques would point out, including those by Timothy Bromage, these changes were not neutral adjustments, but had measurable consequences, with the repositioning of the face leading to a significant reduction in estimated brain volume from 750 down to closer to 500 cubic centimeters in some recalculations. A difference of roughly 30%, which is not a minor discrepancy, but one that fundamentally alters the evolutionary significance of the specimen. Bromage went further, arguing that the reconstruction violated principles of craniofacial development, suggesting that the resulting configuration was not merely unlikely but biologically implausible, a skull that, in his words, could not have functioned in life. This critique underscores a deeper issue that extends beyond a single fossil, because it raises the question of how much of what is being interpreted as evolutionary signal is in fact an artifact of reconstruction, a product of how fragments are oriented, aligned, and ultimately visualized. The problem is not that reconstructions are inherently unreliable, but that they are unavoidably interpretive, particularly when dealing with incomplete and potentially distorted material, and when those interpretations are influenced, even subtly, by expectations about what early humans should look like. The boundary between observation and inference becomes increasingly difficult to define. The issue of distortion adds another layer of complexity, because the parts of the skull most critical for anchoring the face, the cranial base and adjacent vault, are also among the most susceptible to post-depositional deformation. Compression, warping and fragmentation can alter angles and proportions in ways that are not immediately obvious, yet have significant downstream effects on measurements such as facial projection, cranial height, and overall morphology. If these regions are even slightly distorted, then the reconstructed relationships between the face, the brain case, and the mandible may not reflect the original anatomy, but rather a compromise between damaged structures. This becomes particularly important when considering how these measurements are used to define species boundaries, because distinctions between Homo habilis, Homo rudolfensis, and Homo ergaster are often based on differences in cranial capacity, facial shape, and vault proportions, precisely the features most sensitive to reconstruction choices. If these features can shift depending on how the skull is assembled, then the categories themselves begin to blur, not necessarily because the underlying biology is ambiguous, but because the methodological framework used to interpret that biology introduces variability. When one turns to other fossils from the same region, the picture becomes even more complicated because rudolfensis does not exist in isolation, but alongside a series of other specimens, including ER3733 and ER3883, both of which have been assigned to Homo ergaster, and which are often used as reference points for what a more derived, more clearly defined Homo morphology should look like. These skulls, generally dated to around 1.6 million years, display a different suite of features, including thicker cranial bones, more pronounced brow ridges, and cranial capacities typically in the range of 800 cubic centimeters, creating a contrast with the more ambiguous morphology of Homo rudolfensis. Yet even here, the differences are not always as clear-cut as they are presented, because the range of variation within these specimens overlaps in ways that complicate simple classification. The brain size of rudolfensis, depending on reconstruction, can fall within or below the lower end of the range for these later specimens, while its facial morphology, again depending on orientation, can appear either more primitive or more derived. This creates a situation in which the same fossil can be interpreted in multiple ways, each consistent within its own framework, yet leading to different conclusions about evolutionary relationships. When compared to the Dmanisi fossils, the facial projection becomes even more of a problem, because Dmanisi 5 has a particularly massive face paired with brain case strikingly similar to the Homo ergaster specimens. The introduction of Turkana Boy adds yet another dimension to the debate, because here we have a far more complete skeleton, one that provides insight not only into cranial morphology, but into body proportions, growth patterns, and overall anatomy. Discovered in 1984, this juvenile specimen has often been presented as a relatively unambiguous example of Homo ergaster, a tall, slender individual with long limbs and a body plan well adapted to endurance walking and running in open environments. At first glance, this seems to offer a point of stability, a reference against which more fragmentary fossils can be compared. However, the fact that Turkana Boy is a juvenile introduces its own set of challenges, because growth and development can significantly alter craniofacial proportions over time, meaning that the relationship between the face, the jaw, and the cranial base in a young individual may not reflect the adult condition. This complicates comparisons with adult skulls, because differences that appear taxonomically significant may in fact be reflecting stages of growth rather than distinct species-level traits. Moreover, the reconstruction of Turkana Boy is not immune to the same issues that affect other fossils, particularly in the positioning of the mandible and the orientation of the face. Some critiques have suggested that certain reconstructions place the jaw in a position that would reduce airway space or create biomechanical inconsistencies, raising questions about whether the assembled skeleton accurately reflects a living organism. These concerns echo the criticisms directed at ER 1470, suggesting that the problem is not confined to a single specimen, but is inherent in the process of reconstructing fragmentary remains. When viewed together, these fossils form a complex and somewhat unstable network of comparisons, in which each specimen is used to define and validate the others, creating a circular framework in which assumptions about one fossil influence the interpretation of another. If rudolfensis is reconstructed to appear more human-like, it supports a model in which larger-brained humans emerge earlier, which in turn shapes expectations for other fossils. If, however, it is reconstructed in a more primitive configuration, the timeline shifts, and the apparent diversity of early humans may begin to collapse into a smaller number of more variable populations. This is where the rudolfensis controversy intersects with broader questions about human evolution, because it highlights the extent to which the narrative is built not only on the fossils themselves, but on the methods used to interpret them. The distinctions between species, the timing of key transitions, and the identification of ancestral lineages all depend on the reliability of these reconstructions, and when that reliability is called into question, even in specific cases, the implications extend far beyond the individual specimens. It is important to recognize that these debates are not occurring on the fringes of the field, but within its core, involving researchers who are deeply embedded in the study of human evolution, and who bring different perspectives and methodologies to the same set of evidence. The disagreement is not simply about whether rudolfensis belongs to Homo rudolfensis or Homo habilis, or whether Turkana Boy represents a typical Homo ergaster, but about how we move from fragmented, often distorted fossils to coherent biological interpretations. In this sense, the controversy serves as a reminder that paleoanthropology is not merely a process of discovery, but of reconstruction in the fullest sense of the word, a discipline in which the past must be assembled piece by piece from incomplete and sometimes ambiguous evidence. The fossils do not speak for themselves, they are given voice through the interpretations placed upon them, and those interpretations are shaped by both data and expectation. As one continues to examine the details, the angles of the face, the thickness of the vault, the estimated volume of the brain case, the positioning of the jaw, it becomes increasingly clear that small decisions can have large consequences, not only for how individual fossils are classified, but for how the broader story of human evolution is constructed. What appears at first to be a straightforward progression from more primitive to more derived forms begins to reveal itself as a more complex mosaic, in which variation, uncertainty, and methodological choices play a significant role. And perhaps that is the most revealing aspect of the rudolfensis controversy, not that it provides a definitive answer, but that it exposes the process itself, the way in which scientific narratives are built, challenged, and revised over time. It invites a different kind of attention, one that looks not only at the fossils, but at the assumptions that underlie their interpretation, and asks whether those assumptions are as secure as they appear. Because if a skull can change its classification depending on how its fragments are aligned, if its brain size can shift by hundreds of cubic centimeters based on reconstruction, if its facial structure can move from primitive to modern with a slight adjustment in angle, then the line between species becomes less a fixed boundary and more a moving target, shaped by the interplay between evidence and interpretation. And in that space, between what is found and what is inferred, between bone and narrative, something does not quite settle, leaving open the possibility that the story we tell about these fossils is still, in important ways, unfinished.